Thank you. Thank you, everyone. Can you all see my screen?
Yep.
Okay. Yes. Thank you. So I'm reporting my PhD dissertation research, clinical and cost effectiveness of systemic treatments for advanced prostate cancer, network meta-analysis, and economic evaluation. So I'll introduce my research by this outline. I'll introduce background first, then objectives, 3 aims, and finally discussion. So first, some background. So prostate cancer is the most common cancer among men in the United States. About 1 in 8 men will be diagnosed with prostate cancer through their lifetime, and it's the second leading cause of cancer deaths in men. About 1 in 42 American men will die of prostate cancer. Currently, there are more than 3.7 million living cases, and it is expected that in 2021 there will be 248,000 new cases and 34,000 new deaths. So around 80% of the patients were first diagnosed with localized prostate cancer, which is confined to prostate. Beside active surveillance, radical prostatectomy, or radiation therapy can be given with the intent to cure. However, around 30% of patients experience biochemical recurrence, indicated by a rising serum level of PSA, prostate-specific antigen, and require salvage local therapy. As prostate cancer relies on antigen for proliferation, lowering serum antigen levels by medical or surgical castration, also known as antigen deprivation therapy (ADT), can initially control disease progression. Over time, most prostate cancer develop resistance to castration. In some cases, cancer metastases can develop early in castration-sensitive stage. In others, cancers become castration-resistant but remain metastasis-free. Ultimately, patients succumb to metastatic castration-resistant prostate cancer, which has a dismal 5-year survival of 30%. Therefore, recent research has been focused on treating metastatic castration-sensitive and non-metastatic castration-resistant prostate cancer, delaying them from progression to the final stage. Research has shown that the progression of prostate cancer despite castration's serum level of testosterone is driven by persistent androgen receptor signaling. New treatments that further inhibit androgen signaling pathway have been introduced for metastatic castration-sensitive and non-metastatic castration-resistant prostate cancers. These treatments belong to 2 types, hormone therapy and chemotherapy. Hormone therapies can be categorized into 2 classes. One class is androgen receptor antagonist. They include enzalutamide, apalutamide, and darolutamide. Darolutamide is structurally distinct from enzalutamide and apalutamide and has lower penetration of the blood-brain barrier. Therefore, it has fewer central nervous system-related adverse events such as seizures or dizziness. The other class is androgen synthesis inhibitor, abiraterone acetate. It can increase steroid precursors and needs to be given with glucocorticoids. In terms of chemotherapy, docetaxel has been introduced for metastatic prostate cancer. It is a taxane that inhibits mitosis and androgen receptor nuclear translocation. Despite different mechanisms of action, these treatments have undergone parallel development through a series of clinical trials. The availability of these drugs has improved prostate cancer outlook in the meantime, the multitude of treatment options has complicated decision-making, as there is a lack of clarity in comparative effectiveness compounded by a wide range of drug costs. The unclear comparative effectiveness is reflected by the hesitance of clinical guidelines to recommend one drug over the others. If we take a look at National Comprehensive Cancer Network NCCN guideline for metastatic castration-sensitive prostate cancer, several treatments are listed as category 1 recommendation, which means based on high-level evidence, there's uniform consensus that the intervention is appropriate. Similarly, for non-metastatic castration-resistant prostate cancer with high risk of progression, defined by a prostate-specific antigen doubling time of 10 months or less, several treatments are listed as category 1 recommendation. Here we used NCCN guidelines as an example. In fact, we found that uniformly across different clinical guidelines.
First, Stephen, it's talking about prostate cancer. You're back?
No, I'm going.
Oh, you're going. Okay. All right, you can leave the door open, and if you're going.
So in fact, we found that uniformly across different clinical guidelines, the recommendations were made without clear preference. So in addition to unclear comparative effectiveness and safety, the drug costs vary widely. For metastatic castration-sensitive prostate cancer, the drug acquisition costs for patients to complete all planned courses of treatment range from around $300 for docetaxel to about $260,000 for apalutamide. Similarly, for non-metastatic castration-resistant prostate cancer, we can see that the price for the drug cost for a patient to complete or planned courses of treatments range from $9,700 for abiraterone acetate to about again $260,000 for apalutamide. So we will want to ask, are higher drug costs justified by better effectiveness or safety profile? Therefore, the overarching goal of our research is to compare the clinical and cost effectiveness of systemic treatments for advanced prostate cancer through network meta-analysis of clinical trial data and economic evaluation. So here are our specific aims. So for aim 1, we want to assess the comparative effectiveness of metastatic castration sensitive prostate cancer systemic treatments. We use randomized controlled trials data, mainly published aggregate data, and the method we use is network meta-analysis. For aim 2, we would like to assess the comparative effectiveness of non-metastatic castration-resistant prostate cancer systemic treatments, and our data sources are from both randomized controlled trials and single-arm trials, and I'll explain why we use single-arm trials in a minute. And we'll use both patient-level data and published aggregate data. And the methods we used are matching-adjusted indirect comparison and network meta-analysis. For aim 3, we assessed cost-effectiveness of systemic metastatic castration-sensitive prostate cancer treatments, and our data sources were from the M1 analysis, the cancer registry, and claims data. Use both published aggregate data and patient-level data. The method we used is partition survival decision analytical modeling. So for aim 1, we assessed the comparative effectiveness of systemic metastatic castration-sensitive prostate cancer treatments. So for metastatic castration-sensitive prostate cancer, continuous androgen deprivation therapy represented the standard of care from 1941 until 2015, when randomized controlled trials showed that docetaxel improves overall survival when added to angiogen deprivation therapy. Subsequently, abiraterone acetate, uplutamide, and enzalutamide were found to be effective in randomized controlled trials and approved by FDA for the indication. So the method we used for AIM-1 is we search for eligible trials from the bibliographic databases, trial registries, and regulatory review packets. The eligible trials are parallel group randomized controlled trials with at least 12 months follow-up. And here's our pie code. So we're interested in metastatic castration-sensitive prostate cancer patients. Interventions of interest are these chemohormonal therapies in combination to androgen deprivation therapy, and our comparative interest was androgen deprivation therapy. Our outcomes of interest were overall survival and radiographic progression-free survival as our efficacy outcome, and our safety outcome of interest was serious adverse events. So we used the Cochrane risk of bias tool version 2 to assess the trial quality. Specifically, we assessed the risk of bias arising from these 5 domains: randomization process, deviation from intended interventions, missing outcome data, measurement of the outcome, selection of reported results. So for each domain, judgment will be based on answers to a series of signaling questions. Judgment can be high, low risk of bias, or can express some concerns. In terms of statistical analysis, we perform network meta-analyses using Bayesian hierarchical generalized linear model with Markov chain Monte Carlo. So briefly, what is network meta-analysis? It is an extension of pairwise meta-analysis to allow comparisons of more than 2 interventions in a single analysis for all relevant randomized controlled trials. The advantages of network meta-analysis are these 3 I briefly listed here. So first, it compares multiple treatments in absence of head-to-head trials. Secondly, it uses direct and indirect evidence to gain greater precision in estimate. So just to show you what is direct evidence, for example, if we want to compare treatment A versus treatment C, we can use direct evidence from randomized controlled trials that have compared treatment A and C. And you can also use trials that have compared, for example, A to B and C to B. So these trials will indirectly inform the relative effectiveness of treatment A and C. By using both direct and indirect evidence, it gains more precision with a larger sample size in estimate. And it produces coherent estimates of relative effects of any 2 interventions, thus giving us the ability to rank all interventions. So we estimate, in our study, we estimate the relative effect between any pair of treatments. So for our efficacy outcomes, overall survival and radiographic progression-free survival, we estimated time invariant hazard ratio. We also estimated time varying hazard ratio using parametric survival network meta-analysis. In terms of safety outcome, we assessed the odds ratio of serious adverse events. As you know, to assess time varying hazard ratio using parametric survival model, you need patient level data. However, we only have published aggregated data. So we were able to construct a time to event data at individual level by from published Kaplan-Meier curves. By finding the numerical solution to the inverted Kaplan-Meier equations using where available information on number of events and numbers at risk. Just to show you the accuracy of this method, we plot our Kaplan-Meier curves on the right and then fit the Cox proportional hazards model as what the investigators did in the original trial, and we came to the same conclusion in terms of the hazard ratio. So the outcome, so the output of this aim has been published on JAMA Oncology. So I use the figures and tables from our publication for this aim. So here are our results. We found 7 randomized controlled trials with about 7,000 patients with a follow-up duration of 14 to 84 months. Here we illustrate the trial network in which nodes represent treatments and edges show which treatments have been compared in RCTs. So the labels on the edges are randomized controlled trials of pairs of treatments. The node size is proportional to the number of trials evaluating each treatment. The edge thickness is proportional to the precision of the comparison. So it's relevant to the sample size. Edges with the gray color represent a multi-arm trial named Stampede. Most treatments have not been compared in head-to-head trials, as you can see, which highlights the importance of this network meta-analysis. Here we present the risk of bias for overall survival. We noticed that some concerns were raised for missing outcome data in 3 trials. In these trials, the number of patients with missing data was more than 10% of the number of events and distributed unevenly between treatment groups. Yet no analysis was done to correct for the bias due to missing data, and no sensitivity analysis was done to show that results were little changed under different assumptions about the mechanism of missing data. Also, some concerns were raised for the selection of reported results. In these trials, the trial protocol and the statistical analysis plan were finalized after the data cutoff date, and unblinded analysis was performed according to the protocol, leading to the reported results. And here we show the treatment ranking and relative effect of different treatments. So as we can see here, for overall survival, abiraterone acetate provided the largest overall benefit with the lowest hazard ratio. For radiographic progression-free survival, enzalutamide provided the largest benefit and with the lowest hazard ratio. So if we consider both overall survival and radiographic progression-free survival, enzalutamide and apalutamide provided the most consistent benefit for both intermediate outcome and final clinical outcome. In terms of serious adverse events, we observed that in docetaxel, the odds ratio of serious events increased dramatically, 23-fold in increasing the odds ratio of serious adverse events, mainly due to its chemotherapy, and it can induce neutropenia and fever. And slightly increased series of adverse events were observed in abiraterone acetate, which needs to be used together with corticosteroids. And here are results based on time-varying hazard ratio. So we plotted the expected survival curve for each treatment, and the treatment ranking and relative effect were consistent with the time invariant hazard ratio results. For aim2, we compared the effectiveness of systematic non-metastatic castration-resistant prostate cancer treatments. For non-metastatic castration-resistant prostate cancer, the progress in treatment has been slow until 2018, when FDA approved the first drug, apalutamide, for the indication based on randomized controlled trials showing that apalutamide delayed metastasis when added to androgen deprivation therapy. Subsequently, enzalutamide, darolutamide were approved by FDA for the indication based on randomized controlled trial evidence. In addition, a phase 2 single-arm trial named Imogen showed that patients treated with abiraterone acetate plus androgen deprivation therapy had a similar metastatic-free survival to patients treated with other FDA-approved drugs. And as just mentioned, abiraterone acetate has only been tested in this single-arm trial. Because of the comparable metastasis-free survival benefit observed in this trial, some suggest that we should recommend abiraterone acetate in non-metastatic castration-resistant prostate cancer patients. And although the manufacturer of this drug sponsored the phase 2 trial, which showed promising results, it is not pursuing a phase 3 trial for the indication because the patent of the drug expired in October 2018 due to its failure in a generic challenge, and the generic version of abiraterone acetate is now available in the US market. So in order to compare these treatments for non-metastatic castration-resistant prostate cancer, we searched not only randomized controlled trials, but also single-arm trials with a different set of interventions from before the different indication. For efficacy, we assessed metastasis-free survival instead of radiographic progression-free survival, as these patients were initially metastasis-free. So the methodological challenge for this aim is that network meta-analysis compares treatments through common comparators. It necessitates a connected network. It is insufficient when there are disconnected nodes. That is when some treatments were only tested in single-arm trials. So we need to use a different statistical analysis tool in our toolbox, a method called matching-adjusted indirect comparison. This method was introduced by the National Institute for Health and Care Excellence, NICE, in UK. It is used in a scenario where a company has individual patient trial data for its own product, but only published aggregate data for its competitor's drug. In order to compare the two products, the method uses available individual patient data to account for between trial imbalance in terms of baseline characteristics. So suppose we have aggregate data for study A and individual patient data for study B. This method reweights the study B population such that the weighted population has matched baseline characteristics with the study A population. The mean outcome of weighted study B population can then be compared with the mean outcome of the study A as if they are estimated from the same study. The weights assigned to individuals in study B is equal to the odds of being enrolled in study A versus study B. This method uses methods of moments to allow we only have individual patient data for one study, but aggregate data for the other. So we requested individual patient trial data for this single-arm trial Imogen from the YODA project. And here's our trial network. We identified 6 trials with 4,000 patients comparing 6 treatments with a follow-up duration range from 18 to 40 months. From the trial network on the left, we can see that abiraterone acetate was only tested in this single-arm trial, Imogen. Using matching-adjusted indirect comparison, we were able to match the Imogen trial to the Spartan trial, which is a trial with similar distribution of baseline characteristics before matching, and a better balanced baseline characteristics after matching. And we were able to create a pseudo randomized controlled trials, if you will, using matching adjusted indirect comparison. And with this pseudo RCT, we successfully connected the dots and performed the network meta-analysis. So here we present the risk of bias within trials using a different outcome. So before when we presented the AIM-1 results, we presented overall survival. Here we presented the metastasis-free survival, an outcome that requires subjective judgment. There are additional concerns for risk of bias. So besides concerns for missing data and selective result reporting introduced in AIM-1, for AIM-2, concerns were also raised for deviation from intended intervention because it may due to the awareness of assignment by participants or caregivers, because we have single-arm trials and unblinded RCTs. And the efficacy analysis was not based on intention to treat population. Concerns were also raised about the measurement of the outcome, again, because of the outcome assessors being unblinded, and the outcome assessment could have been influenced. And concerns were also raised for the randomization process because single-arm trials were subject to selection bias. Here we presented results of treatment ranking and relative effect. You can see that for metastatic-free survival, abiraterone acetate, although not approved for the indication and only tested in the single-arm trial, provided largest benefit in terms of metastatic-free survival, has the lowest hazard ratio. And for the overall survival, because the single-arm trial for abiraterone acetate did not test overall survival, we did not include this abiraterone in the overall survival treatment effect comparison. And we were able to compare the approved treatments, and among the approved treatments, darolutamide was associated with the largest overall survival benefit. And if we look at the serious adverse events, we can see that darolutamide again is associated with the lowest serious adverse events odds ratio as compared to a placebo. And abiraterone acetate was associated again with slightly increased serious adverse events as before for its approved indication, metastatic castration-sensitive prostate cancer, due to its combination with corticosteroid. And here are the expected survival curves, again, based on time-varying hazard ratio, and it's consistent with the treatment ranking, assuming time-invariant hazard ratio. So for AIM-3, we assess the cost effectiveness of the systematic metastatic castration-sensitive prostate cancer treatments. So here are some parameter setting for the economic evaluation. So, we're interested in metastatic castration-sensitive prostate cancer patients receiving treatment in cancer centers, and we took the U.S. healthcare sector perspective and a lifetime horizon, which is 30 years, after which the mortality reached 100%. And here we specifically listed the treatments of interest and their regimen. We used partition survival model to model disease progression and treatment effectiveness in a cohort of simulated metastatic castration-sensitive prostate cancer patients as they experience 3 different health states: pre-progression, post-progression, and death. The partition survival model is the most commonly used decision analytical approach for the appraisal of interventions for advanced or metastatic cancers. It has been used extensively in NICE in UK. The proportion of patients in each health state after each model cycle of 1 month was determined by the progression-free survival and overall survival curves. Specifically, the area under the progression-free survival curve indicates the proportion of patients remain pre-progression, progression-free over time. And the area above the overall survival curve indicates the proportion of patients deceased over time. The area between the progression-free survival curve and overall survival curve indicates the proportion of patients in post-progression state over time. So here the survival curves for individual treatments were derived by applying the time-varying hazard ratio estimated from M1 to the reference survival curves of androgen deprivation therapy alone arm. And the reference overall survival curve for androgen deprivation arm for androgen deprivation therapy alone arm was derived by synthesizing both trial data and cancer registry data. Specifically, we calibrated the trial data with SEAL data to improve survival extrapolation beyond trial duration. We assembled a cohort of trial-eligible SEEK population, followed up till the chemohormonal therapy introduction in 2015. And we allow the trial and the SEEK survival curves to have different scale and shapes. So here in this graph, I'm showing, so as you can see, these short Kaplan-Meier curves, stepped curves are from trials, and by synthesizing from trials, are from the ADT alone arm of trials, and we're synthesizing these trial arms and to fit the model. So this is the trial ADT alone arm survival curve from trials. And this longer Kaplan-Meier curve here is from SEEK, from external cancer registry. And we also fit a model to model this survival curve from SEEK. We allow these two curves to have different scale and shape. So we respect the heterogeneity between the trial population and SEEK population while allowing the SEEK data to inform the long-term survival of the trial data extrapolation. So in terms of costs, we have included treatment-specific costs, health state costs, and adverse events costs. So for treatment-specific costs, we included drug acquisition costs and administration costs. Specifically, drug acquisition costs were from the Veteran Affairs Department Federal Supply Schedule. The administration costs were from Medicare Physician Fee Schedule. The health state costs were estimated from market scan claims data with Medicare supplement. We included costs incurred in health system net of the treatment-specific costs. For adverse events cost, we used the healthcare costs and utilization project data from AHRQ, the Agency for Healthcare Research and Quality. We derived adverse events probability based on FDA drug label, including both trial data and observational data. And we included grade 3 to grade 4 adverse events that happen in more than 0.5% of the patients, considering their clinical implications and cost implications. In terms of utility, so the health-related quality of life was measured by a utility score on a scale of 0 to 1, whereas 0 represents death and 1 represents perfect health. So the utility scores differ by health stage. So pre-progression or post-progression patients, they will have different health utility score, and it will decrease due to adverse events. So for the health state utility, we use published literature. The method for estimating the utility is the EQ5D quality of life utility score. And for adverse events, we also use published literature. There are different methods we use to estimate the disutility due to adverse events, including EQ5D, time trade-off, and standard gamble. So to assess the cost effectiveness for different treatments, first we need to assess the health outcome, which is the quality adjusted life year. It is simply the length of life multiply the utility in that during the time spent in that state. And to assess cost effectiveness, we assess the incremental cost effectiveness ratio, which is the incremental costs divided by incremental QALYs. And this is comparing 2 different treatments. So basically how many QALYs, so how much more dollars you spend by gaining one quality adjusted life year. And we assess the results both in terms of base case and sensitivity analysis. In base case, the model parameters took mean values. In sensitivity analysis, the model parameters were sampled from distributions. For example, in terms of treatment effect, we used the Bayesian Markov chain Monte Carlo output from the network meta-analysis. For costs, we applied the gamma distribution, which is a skewed right skewed distribution with long tails, and is commonly used for costs, and is strictly positive. For utilities, we used truncated normal bounded between 0 and 1. So here are results. So this is the cost effectiveness efficiency frontier. We put all treatments here with different colors. So the y-axis is the costs and x-axis is the quality adjusted life year. And we want the treatment to be associated with higher costs and higher QALYs and lower costs. And so if you look at the purple dots, these are from enzalutamide. So we can see that it's above the efficiency curve because it incurred, so with the same amount of QALYs, it incurred a higher cost. Therefore, it has been dominated by better treatments. And it is called efficiency curve because each treatment is associated with better QALYs and higher costs all the way up. And here we show the results in a table. So we order the treatments by their quality adjusted life, QALY gains here, and their corresponding mean costs. So androgen deprivation alone arm is our reference treatment. As we introduced, enzalutamide was dominated by docetaxel. As you can see, the 2 treatments has basically the same QALYs, but docetaxel has a much lower cost. And compared to docetaxel, abiraterone has an additional QALY gain, but with additional costs. And the incremental cost effectiveness ratio is $58,800. And apalutamide compared to abiraterone acetate is associated with additional QALYs at a much higher cost. And this is the incremental cost effectiveness ratio, 1.9 million basically for 1 QALY. This is dollars per QALY. So incorporating the probabilistic sensitivity analysis, we were able to show the probability of being most cost-effective against different willingness to pay threshold. We can see here that if someone is willing to pay, so if the health system is willing to pay $50,000, below $50,000, docetaxel is more cost-effective. But with the higher willingness to pay threshold, abiraterone acetate is preferred. The other two treatments, apalutamide and enzalutamide, was never preferred. And in the U.S., the commonly used willingness to pay threshold is 50,000 to 100,000. Finally, discussion. So here are my main findings from the 3 aims. The first aim for metastatic castration-sensitive prostate cancer, we found that abiraterone acetate and apalutamide may provide the largest overall survival benefits with relatively low serious adverse events risk. For aim 2, comparing non-metastatic castration-resistant prostate cancer treatments, we found that among approved drugs, darolutamide has the optimal efficacy and safety, and it's expected as it has lower penetration of blood-brain barrier and lower central nervous system as adverse event. And also abiraterone acetate, currently indicated for metastatic prostate cancer, offered non-metastatic castration-resistant patients metastatic-free survival benefits comparable to the approved drugs, with cost saving from generics. Finally, for aim 3, for metastatic castration-sensitive prostate cancer, we found that abiraterone acetate is the most cost-effective in treatments at the willingness to pay threshold greater than 59,000, and docetaxel is the most cost-effective drug at a willingness to pay threshold lower than $59,000. So our studies have some strengths and some limitations. In terms of strengths, we compared and ranked all recommended treatments for advanced prostate cancer based on all available evidence, including single-arm trials. We were able to use individual patient data and published aggregated data. Besides time-invariant hazard ratio, we were able to model time-varying hazard ratio and more closely model the survival curves. In addition, in the cost-effectiveness analysis, we were able to include cancer registry data for survival extrapolation and claims data for cost calculation. In terms of limitation, some trials, for some treatments, the overall survival data are immature, so it may bias against these treatments. So our analysis will need to be updated with upcoming trial results, so with new trial results. And subgroup analysis was not feasible for metastatic castration-sensitive prostate cancer treatments due to inconsistent cutoff. However, we were able to conduct subgroup analysis for non-metastatic castration-resistant prostate cancer. And if I have more time, I want to introduce the results. The third limitation is that individual patient data were not available for all trials. Ideally, if we have patient-level data for all trials, we can better control for effect measure modification across trials. However, with the current trial data sharing status, we did try it, but we failed to acquire all the patient-level data, trial data. And finally, as we introduced some trials had missing data problem, and some trials have crossover after the primary analysis was done. Patients in placebo group were allowed to cross over to the treatment group and diluted the treatment event. And although there are methods to account for that with individual patient level data, trial investigators did not provide the adjusted results. The results adjusted for crossover. Our study have two major implications. First of all, considering the healthcare burden of advanced prostate cancer and the widely varied treatment costs, this study can inform clinical decision-making and insurance policy to help better align drug costs with their value to patients. Methodologically, this study can augment the capability of network meta-analysis to include treatments with only single-arm trial evidence to address unmet decision needs, because for cancer drugs, 20% of the pivotal trials that supported FDA approvals were single-arm trials. Thank you for listening. Welcome any questions.
Thank you, Lynn.
Thank you.
If anyone doesn't have any, I'll start off.
I have one. Lynn, this is Janet. And is really that your second AIMS finding that a drug that's not approved for that particular indication offers better progression-free survival than the drugs that are approved for it?
Yes. Yeah, that's correct. Because the so FDA did not approve this drug because this drug has only single-arm trial evidence, and it's a phase 4 single-arm trial. It's manufactured by Johnson & Johnson, and after the patent expiration, the company just did not pursue a phase 3 trial because it has another drug. It's also manufactured by Johnson & Johnson, so basically it's promoting the other drug that is still under patent protection. However, abiraterone acetate was clinically used for this indication, so unapproved indication, and so we have claims data papers proving that around 23% of non-metastatic castration-resistant prostate cancer patients were using abiraterone acetate. So clinicians are prescribing this drug basically out of the indication approval.
Wow. Well, what do you think should be the next steps in trying to pursue whether those findings are valid?
So ideally, if there is a NIH-sponsored phase 3 randomized controlled trials comparing abiraterone acetate to, say, ADT alone, or even the other active treatments plus ADT, that would show this drug actually can improve metastatic progression-free survival and maybe overall survival even, because it's indicated for metastatic prostate cancer. So it's reasonable to, and given its phase 2 single-arm trial data, it's reasonable to suspect that it's also effective in non-metastatic setting.
Well, thanks. And I forgot to say, what a tremendous presentation. Really enough for several talks, but congratulations. That was a lot of hard work.
So
thank you, Dr. Holbrook. Thank you.
Very professionally presented.
And that was a great job. And in answer to Janek's question, it's not quite as screwy as it may sound. For example, in ophthalmology, bevacizumab is not FDA approved for the treatment of macular degeneration, but it's every bit as good as the approved drug ranibizumab and 1/8 the cost. And the reason is that bevacizumab is made by the same company that makes ranibizumab, and they want you to only use the more expensive drug. Despite that, there's more widespread use of bevacizumab than ranibizumab. So Whether or not a company goes for a phase 3 trial and approval is based on business decisions, not necessarily whether or not it's going to be the best choice for patients.
Well, I'm familiar with that example, but they at least have the CAT trial to guide treatment decisions. And it seems like something like that is would be a good idea here for NIH to sponsor a head-to-head comparison.
Yeah, yeah, I would hope so.
It was just shocking to me, not because I wasn't thinking so much maybe about the commercial ramifications.
Yeah. Yeah. Um, I, I mean, depending on how long the trial takes. The other drugs, patent are going to expire in 2017, so in 5 to 6 years. So if we can get results earlier, because some trials just have 2 to 3 years of follow-up and show overall survival benefit, we can still have cost saving for the health system and patients.
Lynn, I had more of a methodology question. For your aim to the matching method that you use, the matching adjustment, what are some biases that you're concerned about and how did you control for them?
Yeah, so yeah, and I probably skip that. So in our case, we used yeah, I didn't include it. So in our case, we used age, the years, the duration of prostate cancer diagnosis, and also PSA baseline PSA level, and PSA doubling time, to control for we controlled for patient characteristics, these patient characteristics between the two trials. However, there are residual confounding. There are probably residual confounding because we can control for all baseline characteristics. And I remember, as Dr. Holbrook has mentioned in my proposal, so we assume that the underlying correlations among different baseline characteristics are the same between the two studies. That's an assumption. Yeah.
Okay, thanks. Any other
Well, I
Yeah.
would ask a question related to that. Just, I have two questions to make sure I understood your talk. If you go back to that slide that you were just on.
Which one?
Yeah, this one. So this was allowing you to incorporate a single arm study.
Mm-hmm.
And was it kind of the basis of what happened was you kind of tried to get, it's like, I forget if it's if it's the age adjusted mortality or the standardized mortality, but projected what they were observed to live, and then in the other trials that they weren't actually in, what the patients with those characteristics were observed to survive.
Yes.
And
yeah.
Did that comparison.
Yeah. So basically it's saying, what if the patient were in that study? What if patient, let's say patient B population were in what if patient in study B population were in study A? What if they have similar characteristics? What would we observe in this weighted study B population that looks very much like study A population?
Yeah, well, that seems like, the sort of the standardized mortality adjustment a bit too. And the other thing wasn't sure if I quite understood, could you go to your graphs on the SEER data? Yeah. So is the line that you have for, it's kind of more blue. Okay, so just to get established, the lowest line is the dark green one. That's the SEER, actual SEER data, or?
So yeah, so these two very long curves are modeled curves, and these short lines are these lines are trial data, and this line that is slightly longer than these lines are seek data, because seek has longer follow-up than trials.
So what I assume from this, that all of these trial participants, including the one that the model trial, had better survival than prostate cancer, people with prostate cancer in general.
Yeah, exactly. And actually, we were discussing that with a health economist in health policy department, and we want to maybe publish research letters for that. Like, is like trial population really represents the real world setting? So we assemble a cohort of trial eligible population, but the distribution of this population, like age distribution, race distribution, it might be different from the trial population because it's only trial eligible, but for example, age groups, the distribution may be slightly different or largely different. So that may, in a way, to some extent, explain the difference we see here, why Sikh patients perform worse than trial patients. However, I did another analysis, which I didn't show here. So I used the Sikh data and the charter trial. So one of these trials, and I tried to reweight Sikh population to make it look like chartered population. However, even after the weighting, the Sikh population performed worse than trial. So the curve was shifted upward a little bit, but still worse. So like, and then when we consider it, so the trials were conducted in like large cancer centers. The Sikh population, so even with the same age, same disease duration, same PSA, they may receive care in like less ideal setting. That may be one of the reasons that we observe the difference.
Yeah, and maybe it's obvious that, you know, people in clinical trials tend to do better, but I'm glad I understood your slides.
Yeah, so like, so we want to borrow information from the Sikh population, and on the other hand, we also want to admit that these two populations are different. So we allow these two curves to have different shape and scale. So by including different shape and scale, this longer Kaplan-Meier curve can contribute some information to the modeling of this trial curve. So basically, we use their difference observed during the trial duration to extrapolate how these two populations would be different moving forward. So calibration.
That's good. All right, we're a little bit over, but if there's any other questions, we can take them. Good. All right, thank you, Lynn. Do you have any questions for us before we break?
Any suggestions, like presentation slides? Does anything not clear? I'm planning to defend in March, by the end of March. In the end of March.
Sounds good.
Great.
Good.